Hi everyone. In our last video, we looked at the concept of summation. Now we'll apply this concept to the features of the photoreceptors found in the retina. Photoreceptors are light-sensitive cells found in the retina, of which there are two types, rods and cones. Rods account for about 95% of the photoreceptors and are found distributed throughout the retina, except for the blind spot where the optic nerve connects. Cones are less numerous and are mostly concentrated to a small area on the back of the eye known as the fovea. Photoreceptors contain a pigment which is bleached by light, and then these chemical changes ultimately lead to a change in membrane permeability to sodium ions. As we've seen earlier, this change in membrane permeability leads to a generator potential, which, if sufficient to reach threshold, will initiate an action potential in bipolar cells, and these then synapse with ganglion cells, which are sensory neurons of the optic nerve. Rods exist as only one type, and therefore provide us with monochromatic vision, but can also function in low light intensities, and so it's these photoreceptors we rely on for night vision. In contrast, cones provide trichromatic vision, as there's three types, each sensitive to light of different wavelengths. The three types are blue light sensitive, green light sensitive, and red light sensitive, and so the combination of these provides us with colour vision. Rods are very sensitive to light. This means they can initiate action potentials in the ganglion cells in dim light, and so, as we've already said, provide us with night vision. This ability is due to many rod cells synapsing with a single bipolar cell, so many generator potentials are combined to reach threshold in the bipolar cell. This is an example of spatial summation. Cones are less sensitive to light. Each cone cell is connected to its own bipolar cell, and so only temporal summation will allow for threshold to be met. This would require a strong stimulus, which in this case is a high light intensity. Visual acuity is the ability to distinguish between two close points to produce a clear and crisp image. Rods provide low visual acuity, as several rods are connected to the same bipolar cell. The brain cannot distinguish exactly which rod cell was stimulated. Cones, however, are able to provide a high visual acuity, with each cone cell being connected individually to its own bipolar cell. The brain can interpret exactly which cells have been stimulated. So now we come to a final comparison of rods and cones. Rods are found distributed throughout the retina, whereas cones are most concentrated at the fovea. Rods allow us to see shapes and outlines in monochromatic vision, but with cones we can see in colour. Many rods are connected to a single bipolar cell, whereas cones are connected individually. Rods have a high sensitivity to light, but cones have low sensitivity. This is why you can't see colour very well in the dark, and also why you can't see with enough precision to read. You're only relying on your rods. As rods provide low visual acuity, whereas cones provide high visual acuity. So in bright light, we can see a full range of colours and with much better precision. Our next video will focus on the neuromuscular junction, where a motor neuron meets a muscle fibre. You may want to note that this links nicely to the topic on muscle contraction.